Hello everyone, this is Giselle and my topics are cytogenetic effects, multi-hit chromosome aberrations, and kinetics of chromosome aberrations. You can find information on this in Chapter 33, Deterministic Effects of Radiation, pages 455 through 458. So, what is a cytogenetic effect? To start, cytogenetics is the study of the genetics of cells, particularly the cell chromosomes. A cytogenetic effect refers to changes or damages done to the structure of chromosomes in a cell. Typically, this is caused by radiation exposure. Cytogenetic analysis was developed in the early 50s, and since then it has largely contributed to human genetic analysis and radiation genetics. In order for analysis, a culture of the cells is required. The cells are prepared so the chromosomes can be studied. To better understand cytogenetics, it is best to understand the composition of the human chromosome to understand how radiation can affect the chromosomes in the human body. The human chromosome is a long, coiled structure made of DNA and proteins that contain genes, which are the instructions for building and running the body. A chromatid is one half of a duplicated chromosome that contains DNA to be passed to another cell. As you can see in this photo, it contains a chromosome, and the A side is displaying a two-chromatid structure going through S phase, which is where the DNA molecule is replicated into two identical daughter cells. You can see that A ends in a four-chromatid structure by looking at figure B. You can see where the arrows are pointing where the centromeres are in the chromosomes. A centromere is the region that holds the sister chromatids together. They also help with movement of chromosomes during cell division. All of this will become clearer as we move along in the slides, and their importance will become apparent. The image in this photo is displaying an irradiated human cancer cell. In the photo, I will be introducing you to chromosome aberrations. The labeling on the photo are different types of chromosome aberrations that we will be exploring further. These aberrations are representing a high degree of damage done to the cells. Attempts to measure chromosome aberrations in patients after diagnostic X-ray exams have been largely unsuccessful. And the reason behind this is because diagnostic X-rays tend not to expose an individual to high enough radiation doses to produce such effects in the cells. However, instances of high-dose fluoro studies have shown chromosome aberrations shortly after exposure. The rate of chromosome aberrations is related directly to the radiation dose one is exposed to. What does all this mean? Let's put everything together to make more sense of the effects. To begin, a chromosome aberration is a change in the normal structure or number of chromosomes, and this can lead to many different adverse effects such as a genetic disorder, birth defects, or diseases. The chromosome aberration visualized at metaphase consists of a chromosome with material missing from the ends of two sister chromatids and two acentric fragments, and these fragments are called isochromatids. A dicentric chromosome is a normal chromosome that has two centromeres instead of one. This usually happens due to radiation damage or chromosome rearrangements and can cause genetic damage. A ring chromosome is a type of structural chromosomal abnormality where the ends of a chromosome break off and the remaining ends fuse together to form a ring. Now that we have a better understanding, we can look at this photo and now see these abnormalities in a different light. The arrows point out the dicentric and ring chromosomes as well as the isochromatids. There is no question that high doses of radiation cause chromosome aberrations. However, with doses lower than 100 milligram tissue, these same aberrations do not occur. Something that is still very hard to do is to link these radiation-induced aberrations to latent diseases or illnesses. A latent disease is an illness or disease that can stay inactive in the body for a long time before symptoms start to appear. Cells can sustain cytogenetic damage anytime the body is irradiated. It is just easier to distinguish the damage with high doses. The damage that occurs is classified as an early response to radiation. An early response to radiation can be one that occurs in minutes, hours, or even days after exposure. And it isn't considered an early response because if the cell survives, the damage is manifested during the next mitosis after the exposure. And as you may recall, mitosis is the stage in which a cell divides to make two new identical cells. The response is classified as early not because mitosis happens right away, but because the damage shows up in the very first mitotic event, and that is as early as the cell can respond visibly. So before that, it is hard to detect. Human peripheral lymphocytes are most often used for cytogenetic analysis. These peripheral lymphocytes are most often used due to the fact that they do not move into mitosis until they're stimulated in vitro, or this is also known as in a lab. Again, all cells can sustain cytogenetic damage when the body is irradiated, but it is more prevalent in high doses, and although cytogenetic damage occurs at the time of radiation, it could be months or years before damage can be measured. After all this, you may ask, why does it take that long to detect? Well, the simplest answer is that we can only detect damage during mitosis. And chromosomes are only visible during mitosis, so we have to wait for these damaged cells, like the lymphocytes, to go through mitosis to detect it. So if a person is exposed and we don't collect the lymphocytes right away, the damage stays hidden in the non-dividing cells because lymphocytes usually do not divide unless stimulated in a lab. Before I can talk about multi-hit chromosomes, we need to know what a chromosome hit is. When radiation interacts with chromosomes, it could occur through direct or indirect effect, and this results in a chromosome hit. There's both a DNA and a chromosome hit. The DNA hit results in an invisible disruption of the molecular structure of the DNA. Even though it is described as an invisible disruption, it doesn't mean that no damage was done. It just means that it is too small to see it immediately. A chromosome hit, on the other hand, produces a visible derangement of the chromosome. As you can see in the image below of a single hit chromosome, the hit caused a chromatid deletion in only one arm of the chromosome. The result is an arm that is missing genetic material and a chromatid fragment. This is a great image to review because it shows the effects of aberrations at different stages in the cell cycle. When a single chromosome is hit more than once, it becomes a multi-hit chromosome aberration. This is not uncommon, and unfortunately, chromosomes are large targets in the nucleus, being that they are long strands of DNA, so they occupy a lot of space. Because they occupy a lot of space, they are more likely to be hit multiple times as radiation passes through. In the G1 phase, if the chromosome is hit twice and two breaks are formed on the same chromosome, it could break the ends of the chromosomes when this happens, and now it has two open ends. Radiation induces something called stickiness at those ends, and because of this, the two ends join abnormally, forming a circle, as you can see in the photo to the side. A dicentric chromosome, a chromosome with two centromeres, is produced when adjacent chromosomes each sustain a hit and then recombine. As you can see in the photo of the chromosome to the side, the chromosome is abnormally shaped with these two centromeres. Aberrations can also be produced in the G2 phase and require that either A, the same chromosome be hit two or more times, or B, adjacent chromosomes be hit and are joined together, and this is rare. These are rare because they require two specific hits in a short phase, and it typically affects chromatids rather than whole chromosomes. A reciprocal translocation are multi-hit aberrations that require karyotypic analysis for detection. They require karyotypic analysis because there's no loss of genetic material. Instead, it is simply a rearrangement of genes. All genetic codes are available, however, they may be organized in an incorrect sequence. Kinetics of chromosome aberrations simply is just how the chromosome aberrations form, change, and behave over time. At very low doses of radiation, only single-hit aberrations occur. When radiation doses exceed 1 gray in tissue, the frequency of a multi-hit aberration increases rapidly. Single-hit aberrations are produced with a linear, non-threshold dose-response relationship. Multi-hit aberrations are produced following a non-linear, non-threshold relationship. And if radiation dose is unknown, yet not life-threatening, the approximate chromosome aberration is 2 single-hit aberrations per 10 milligray per 1,000 cells, and 1 multi-hit aberration per 100 milligray per 1,000 cells. This just means that when someone is exposed to radiation, but the exact amount is not known, scientists can estimate the dose by looking at their cells under a microscope. If they find 2 single-hit chromosomes out of 1,000 cells, they can estimate the dose to be approximately 10 milligray in tissue. And if they find 1 multi-hit chromosome in 1,000 cells, they can estimate the patient's dose to be approximately 100 milligray in tissue. As a reminder, nonlinear, non-threshold means that a certain level of exposure must be reached before any biological effect occurs. Below this threshold, the body can tolerate the exposure without harm. Linear, non-threshold means that any amount of exposure, no matter how small, can cause a biological effect. The main idea is there is no completely safe dose. In this image, it shows the dose-response relationships for single-hit aberrations are linear, non-threshold, but the relationship for multi-hit aberrations are nonlinear, non-threshold, as you can see by the curve in the photo. Thank you all for your attention. I hope you all learned something with this video, and at this time, I will be welcoming questions.